Hello guys, this is one of also Dr. V lecture series. I'll be covering series of lectures in microbiology, food microbiology, industrial microbiology, some areas of environmental, petroleum, calculation aspect of microbiology, in-depth and as far much as quality control and the rest of them. In this lecture series, we'll be taking a general microbiology to the basic microbiology. The basic topic we are going to cover today or in this section is basic calculation in microbial growth enumeration. And this lesson, the first lesson, which is introduction to microbial growth. In microbial growth, we all know that microbial growth definition has to do with basic key terminologies and terms that must be there, keywords that must be there, and the basic keywords that must always be in any definition of microbial growth is an irreversible increase in microbial population activities, or any activity that you are going to use, or any parameter you are going to use to measure the microbial growth. There must be the keyword irreversible increase in microbial population or biomass. And basically for this lecture, we are in the introductory part of microbiology, so we are going to focus on the three basic microbial system within the environment that is always recurrent and very prominent. And this group of microorganisms are the bacterial group, the fungal group, and the single cell prokaryote. We are going to be looking at the parasite or the protozoans. These are the three major ones we are going to focus for this lecture, which is a preliminary lecture for students' new introduction into microbiology. So having a look at that, we are going to look at the basic dynamics and the process that plays a key role in function of this ecosystem. How does this microbial growth affect the ecosystem? What is the implication of this microbial growth in ecosystem? How does it affect human being and the environment? These are the areas we are going to look into for this lecture. Having said that, let's go to the next slide. The next slide is a continuation of lecture 1, which is serial lecture, which is lesson 1, introduction. We are also now going to look into what is microbial growth. Microbial growth is a process that refers to a population increase of microorganisms, which is basically irreversible, and we have already identified the three group of microorganisms that we are going to be focusing, which is the bacteria, the fungi, and the protozoa. This microbial growth is the irreversible increase of this microbial population or biomass over time. And it is very key. The over time has so many implications that we are going to discuss in the recalculation, such as the generation time and the generation level. Let's leave that one and discuss more of the basic terms. And another basic term we are going to look, what are the fundamental aspect of this microbiology? What are the fundamental aspects of it? What are the key rules? What are the strategic impact that they have in an environment and our everyday life activity that has to do with our quality control, the environment, and the rest of them? In the next slide of the lesson 1, we are also going to look at what are microbial growth. How does microbial growth initiate cell division and replication? We looked at microbial growth is primarily driven by a process of cell division. The cell has to be multiplied. Every organic cell, every living cell undergoes division in order to replicate, and this is not different from microorganisms and microbial cell, as the case may be. Tailoring our lecture or lesson within these three aforementioned microorganisms, the bacteria, the fungi group, and the protozoa, we are going to look at what are the different modes of cell division which this microorganism initiate and accommodate. Why do they tend to be more predisposed to such mode of cell division? What are the factors and parameters that initiate them or encourage them or make them feel that they should take this method of cell division? Is it for survival mode? Is it for proliferation of high number over time? So all their factors, or is it the environmental factor, all these things plays into the reasons or the factors which this microorganism consider, or biological factors which microorganism put into consideration when they initiate into cell division. One of them, or we are going to take the popular three of them to cover the three group of microorganisms we have already identified that we are going to take in the next slide. Moving to the next slide, the most popular and the most common, and perhaps that is the basics of the calculation we are basically used by bacterial group. In binary fission, what we are looking at is binary fission is a common mode of cell division in bacteria, which involves the following step: that the parent cell divides into relatively equal two daughter cells. The word binary means two, so a parent cell divides, splits into two, and the basic organisms that are very common within our environment and the bacterial group is the E. coli. This E. coli is very common in terms of and is a normal flora of our enteric region, and when they undergo this process, that means for every life cycle, the number or the population of the microbial cell increases or doubles into 2. That means if there is 10 E. coli cells in a particular system, and within every 20 minutes it completes a cycle of binary fission, that means every 20 minutes the 10 cells will multiply into 20, another 20 minutes will multiply into 40, another 20 minutes will multiply into 80 in that exponential order. And the exponential order is related in the equation of an index of 2 raised to power n, which we will discuss in the basic calculation. I just want to introduce it now so that we have an idea of it. These are the major ones. And one thing you must also know in binary fission is that most microorganisms undergo binary fission when the nutritional or the environment is very optimal, when there is enough nutrients, when the environment is conducive and comfortable. The microorganism, especially the bacterial group, undergo this level of cell division to have an exponential growth. It's not a survival system. It's when the nutrient is available, everything is in abundance in excess, so they initiate this group. This is very popular among them. The next one we are going to refer to is very peculiar to the fungi, and which is basically we're looking at the yeast group. And this yeast group, we know of the basic yeast group that's involved in this system of cell division called budding is the Saccharomyces cerevisiae. This microorganism is very friendly to human beings. We use it in most of our food processing. Most of our wine and bread a baking system are being produced by this particular organism. And if you look at this and the budding, that means in the budding system, the parent cell, the division in budding system, the parent cell does not divide to two relatively to equal daughter, but an outgrowth from the parent cell, which will now result into 2. Then the daughter cell will now grow over time into maturation. This system of budding is not very comfortable. It's always in a situation where this environment is not optimum, and the system and the microorganism is trying to survive within that environment. So it always tends to go into budding system so that the parent cell will still be matured in order to produce more, while the daughter cell that is produced may likely be able to survive the environment or not. Even if it doesn't survive, the parent cell have the opportunity to produce more because the environment and the nutrient is not optimal. It might not be sufficient, but it is available for the growth to initiate. So this is by fungi group, which is the yeast. Then we take another one, which is the protozoa. I have the schizogony group. Schizogony in protozoa. This schizogony is a kind of reproduction system where the microorganism, which is the protozoans, are now parasitic to the cells. They invade a host cell in order to use the host cell to replicate their process, and this is basically seen in malaria parasites, which is the Plasmodium species, and parasites the red blood cell and the liver cell, and creating a lot of damage, which results to malaria symptoms and sicknesses. So these are the type of cell division we have in microbiology. These are the three common popular one within the three group of microorganisms we are going to be considering in this lecture. One, first is the binary fission, second is the budding in yeast, which is the fungi, and the third is the size schizogony in protozoa. The last one is a kind of growth in which microorganism adopts when they are in survival mode, when the environment is so stressful and very antagonizing. They tend to go into this mode of growth in order to safeguard their survival and their species over time, even though there is not enough nutrient and the nutrient is depleting and the nutrient is not available or completely absent, or the environmental condition is very hostile and harsh, which may result to death. In order to safeguard the generation, in order to continue the existence of the generation, they tend to go into this spore forming growing system, which is called the endospore. If you look at this, when microorganisms have a survival mode or a harsh environment and initiate this spore formation, the spore formation does not actually go into production or growth. Basically, what it does, the parent cell, in order to initiate a new daughter cell, encapsulates in that spore cell, encapsulate like a protective coating for the daughter cell. If you look at this diagram here, this is the parent cell. When the parent cell is in a vegetative cell, that is when the environment is conducive, it can undergo different binary fusion and other types of cell division to initiate growth. But when the environment is very harsh, this parent cell here will now initiate a protective coating and releases the daughter cell in a capsulated form known as the spore. This spore prevents the harsh environment from eliminating the daughter cell and giving it a chance of survival. Some of them can survive in this spore as much as 1,000 years until the environment becomes favorable. When there is enough water, when there is enough nutrient, when there is the right temperature, they will now move from this spore cell back into the vegetative cell and continue the life cycle of the microbial existence. So these are the kind of growth system that microorganisms adopt when they are in survival mode. Having said that, we now look at what are the group of microorganisms that are basically known to be in the survival mode. One of them is the Clostridium botulinum, and this Clostridium botulinum is one of the major food poison causative agents initiate their growth. And this ends the lesson 1, and in the lesson 2 we are going to discuss more extensively. I'm trying to find a way not to move into a lot of discussion about this area, because the major lesson, the takeaway lesson of this series is basic calculation in microgrowth and microbial enumeration. So let's move to the next one so that we can provide a basic preamble. It becomes more easy. So thank you as we move to lesson 2. So the next video will show you lesson 2. Thank you.